Hello, okay, everybody. Hello, welcome back to the third session of the ABX meeting. Thanks to all the speakers so far. We've got a great lineup for the third session about to kick off. And we're going to have a little bit of a change of schedule and kick off with Kirsty Smitten, who is a PhD student from the University of Sheffield. Her talk is looking at novel ruthenium antimicrobial compounds. So yeah, looking forward to hearing this one.
Hi everyone, I'm Kirsty Smitten from the University of Sheffield, and obviously I'm going to talk to you today about two novel ruthenium antimicrobial complexes that I've developed during my PhD. So the antimicrobial resistance is already responsible for 700,000 annual fatalities, and failure to address the issue by 2050 could result in 10 million deaths a year, costing a 66 trillion loss to the global economy. To make matters worse, a new class of antimicrobials hasn't reached the clinic in over 30 years. Our solution is to generate two new classes of antimicrobials, two lipophilic series, so a dinuclear and a mononuclear series. And here are the lead compounds that we've isolated from these series. So we've exploited a new chemical space and we have a modular 4-step gram scale synthesis. The way I usually describe it is like Lego. So you have the central building block and then we build these outer ligands onto the outside. We can change these ligands to alter the properties of the complex and to alter their targets within the cell. We've tested both compounds across a range of bacteria strains, but I've only isolated a few key ones today. So we've tested on gram-negative and gram-positive bacteria, including clinical isolates and carbapenem-resistant strains. We see a higher activity for both complexes on gram-negative bacteria compared to gram-positive bacteria. I'll discuss why this is later on. So this is on planktonic cells. We also see a high efficacy on biofilms as well. So we've tested on gentamicin-resistant biofilms for MRSA, Acinetobacter baumannii, Klebsiella, and Strep. And we see an activity of around 8 to 16 micromolar for the dinuclear complex and around 16 to 64 for the mononuclear complex. Interestingly, these complexes themselves are luminescent, so we can directly track where they are in the cell using optical microscopy. And when we look at the biofilms under a microscope, we see that the compound penetrates the biofilm, which is obviously something that clinical antibiotics can't always do. Because we can look at where the complexes are in the cell using light microscopy, I've directly tracked their mechanisms of action and their cellular targets. So for gram-negative bacteria using E. coli, the dinuclear compound initially binds the outer membrane within 5 minutes. So the red you can see is confocal microscopy and the green is stimulated emission depletion microscopy, so super resolution. We think this binding is to lipopolysaccharides on the outer membrane. Within 60 minutes, the compound then localizes at the poles of the cell, which we've attributed to a novel target. So this is undisclosed at the minute, but we've confirmed this with knockout studies and molecular modeling. The compounds themselves are also electron dense enough, which means they can act solely as an electron microscopy stain. So using transmission electron microscopy, we've looked at the cells at 5 and 60 minutes. So at 5 minutes, we see perforation and swelling of the outer membrane. And then at 60 minutes, we start to see the inner membrane peeling away and the cells beginning to lyse. We've quantified this using ATP release assays. So we see a significant increase in ATP release upon incubation with MIC levels of the dinuclear complex. And we also see an increase in membrane potential. We have now also quantified this with qPCR reactions, which we've just published a paper on. For gram-positive bacteria, so for Staphylococcus aureus, we don't actually see the same binding. So we see spotting around the cell wall, which we've attributed to binding to teichoic and lipoteichoic acids. And we know this is the case because I've knocked out deanylated teichoic acid, so delta A, and then wall teichoic acid, so taro. And we see an increase in activity up to the same amount that we then see in the gram-negative bacteria. So what we've proposed is that the reason the complexes are less active on gram-positive strains is because they're binding to these lipoteichoic and teichoic acids, preventing enough of the compound going into the cell. So we do see less accumulation of the complex in the gram-positive strains. Interestingly, the mononuclear complexes have a different mechanism of action. So within 5 minutes, they directly target the bacterial DNA. This occurs both in E. coli cells and in Staphylococcus aureus. So the green again is confocal, and then the red is the super resolution. I've done a variation on the Ames test, so the fluctuation method, and we've observed mutagenic effects for the compound within bacterial DNA. I've also done HPRT genotoxicity studies on mammalian cell lines, and both compounds are non-mutagenic to mammalian DNA when compared to a known mutagen such as cisplatin. I've tested the toxicity of the compounds in vitro and in vivo. So I've tested them on HEC293 cells. Also now we've added in skin cells, bladder cells and epithelial cells to our list of our panel of mammalian cells. So for the dinuclear complex, we see a IC50 of around 100 to 180 micromolar, depending on the cell line. And for the mononuclear complex, it's around 80 to 100 micromolar. This gives us a therapeutic window of around 2 orders of magnitude, which is obviously significantly higher than you see for quite a few of the last line antibiotics. I don't have an animal license, so I've used Galleria melanella for my in vivo model. So we went up to doses of 80 mg per kilogram, which works out about 100 times the MIC. On the graphs, you see labeled A, so these are live and dead scoring. So what I do is I inject the larvae with the compound, and then I monitor their live and dead scores for 120 hours. I also monitor their activity. So something that Galleria do when they're under pressure or when they're ill is they melanize and they start to turn black. So I also score this as well. We saw no statistical difference between the controls. So the controls are the black line, and then my compound treated, which is the red line. The reason that we do see some death, even with the controls, we think is because for the next stages, I have to cultivate the bacteria, obviously at 37 degrees. So I incubate these larvae at 37.5 degrees, and they actually like to be at 15 degrees. So we think this is why we do see, even with the controls, some larvae dying. I've used the larvae to do an infection model. So I had to determine the concentration of Acinetobacter baumannii that's pathogenic to the larvae. And what you need is a slow death. So you don't want all of the larvae to die within the first 24 hours, because then we might not be able to monitor clearance of the infection. So we need to get the concentration of A. baumannii that will result in the larvae slowly dying over the 120 hours. So I used AB184, which is the most prevalent clonal group of Acinetobacter baumannii in the UK. And what I do is I infect the larvae with Acinetobacter, and then with 30 minutes later, I'll inject with my compound. So I did live dead scoring as with before, and we see 100% survival when the compounds are treated with both the dinuclear and the mononuclear complex. The good thing about Acinetobacter is it infects the larvae's hemolymph. So Galleria have a hemolymph system, which is a bit like a blood system, and they have hemocyte cells that act like neutrophils and macrophages that can phagocytose bacteria. And you can actually extract this hemolymph, and then you can plate this onto agar and count your colony forming units per mil. So you can see how much bacteria is within the hemolymph, which is really good, obviously, for monitoring clearance of an infection, because you can actually watch the infection clear through plating out the hemolymph. So for the mononuclear complex, the infection clears with a single dose within 72 hours. And for the dinuclear complex, it's within 120 hours. We are going to increase dosing or try different concentrations, but this is how far we've got at the moment. So I've developed a novel mechanism to turn the skin of the larvae clear. So I can't go too much into detail because we haven't actually published it yet. But I trialed, so basically what I do is I do the same infection as I did in the previous slide, and then I put the larvae to sleep. So I trialed using different anesthetics, and I started to see melanization. So I have to put them in a diethyl ether chamber, because this was the best way to get the larvae to not melanize whilst I'm putting them under anesthetic. And then I image the infection. So these are just slices. If you might not be able to see it here, but down the middle is the hemolymph, and the hemolymph cells are phagocytosing the Acinetobacter baumannii that's labeled with the compound. We've done experiments where we label this Acinetobacter with GFP, and we don't see so the larvae can't deal with the infection when it's labeled with GFP, but when we introduced ITMP into the system, the compound is also killing the bacteria, and then the larvae's immune system can then deal with the infection. So we're working on whole larvae imaging at the moment. This is on a light sheet. So this is Acinetobacter labeled with GFP, and then we're looking at it under a light sheet microscope. There's quite a bit of autofluorescence at the moment, so we're having to trying to work with the settings on the microscope and are clearing to change this. We've just bridged the gap between in vivo and in vitro studies by doing an ex vivo efficacy model on porcine corneas. So what we do is we extract the pig eyeball, we grow a Pseudomonas biofilm onto the eyeball, and then treat with MBEC and 2 MBEC concentrations of the complexes, and monitor the clearance of or eradication of the biofilm. So at 2 MBEC concentration from a single dose within 18 hours, we see a 75% reduction in Pseudomonas biofilm. So currently we've just finished our in vitro DMPK work profile, which is looking quite promising. We're developing an ex vivo model similar to the porcine cornea, but with Pseudomonas and primary human alveoli cells. We're hoping to move into a clinically relevant small animal model. And we're also looking at other routes to market. So I'm currently on Innovate UK's iCure program to look at how we can take these compounds to market. And we're looking at potential other applications such as coatings within within stents, catheters, and endotracheal tubes. Thank you for listening. Has anyone got any questions?
Thank you, Kirsty. Thanks for that talk.